Hello students, welcome to Biosastra Talks. I hope all of you are doing well, and as we are going on with the NEET 2025 biology paper here, and next questions we are going to revise here so that you can analyze and see what were the answers. So we'll go on with the Q&A session here from question number 111 to 120 from the biology session. Okay, let's get into the topic. So what was that question? First question given here is given below are the stages in the life cycle of pteridophytes. Arrange the following stages in the correct sequence. So in this question they are asking the sequence, the stages of the pteridophyte life cycle. Okay, if you see here, normally they have given some options. In these options you have to select which is the correct order for the life cycle of pteridophytes. Okay, if you see here picture, we know that there are so many types of pteridophytes. Just for your convenience I have given the picture. You can see that there are some types of ferns like Selenella and etc. as well. So these are all the pteridophyte plants that were seen in the environment. Okay, if you see, if we move on to the life cycle, so to know the answer, so what is the life cycle of a pteridophyte is, if you see the picture here, you can analyze it in this. First of all, that mature pteridophyte, or the you can say these are the sperms. This is the life cycle of a fern life cycle. Okay, so if you see that there is a fern, first of all it will undergo, it will release the sporangiums. Okay, these sporangiums undergo meiosis division first. So by the meiosis we know that each one will be divided into four, right? So through this meiosis process, spores are produced, or the sporophytes are produced. Okay, these sporophytes are then entered into the gametophyte. So sporophytes or spores, and then the formation of antheridiums in the gametophyte itself. You can see that a young gametophyte is formed here. So this is the mature gametophyte. So this mature gametophyte contains some antheridiums like this. So these antheridiums contain antheridia, you can say that sperms. In our language you can say that male gametes. Okay, so these antheridia, they are present in this antheridium. Okay, so these antheridia are then, when they are produced totally, then these antheridia are released into water. Okay, normally we know that the pteridophytes are present in the water, so they are released into the water, and then they'll go and attach to the female archegonium. Means the transfer of, here you can see that the transfer of antheridium into the archegonium in the presence of water. Why? Because it happens in the presence of water. Okay, so you can see the transfer of antheridium into the archegonia, and then at last the fertilization. Means when the sperms enter the egg, you can see the fertilization process and the formation of zygote. So this is the whole cycle of a meiosis process, the life cycle of a pteridophytes. You can see here. Okay, it happens like this only in every pteridophyte. So according to the question, you can give the options, like they have given so many options. First of all, in that options only you can see that meiosis, and then formation of the sporophytes or spores, and then formation of antheridia in the gametophyte. Okay, so the transfer of antheridia from into the archegonium in the presence of water, and then fertilization. So if we see the order from the options, obviously you can say that the correct sequence or the correct option from the question is 1. Okay, option 1, in which you can see the order from the question. So this is the order, and you can, in the pteridophytic life cycle, you can see an alternation of generations. Why? Because you can see both meiotic divisions and mitotic divisions occur in the same life cycle of a pteridophytes. That's why you can say that the pteridophytes undergo alteration of generations. Okay, so normally the sporophytes are diploid, okay, which were produced from the meiosis, whereas the spores are haploid. Spores means sperms are haploid, which were produced by the meiosis. Okay, that's it. We'll move on to the next question. So what is the next question is, it came from the cardiac cycle actually. So if you see the cardiac cycle, what is the question here is, the cardiac activities of the heart are regulated by. Here you have to say that they have given some options. You have to say that which were responsible for the regulation of the cardiac cycle. We know that cardiac cycle includes 4 stages, that is diastole, and normally 2 stages, diastole and systole, and it occurs in 4 steps. Means diastole means the tricuspid and tricuspid and mitral valves will open. Okay, and then relax, and again diastole and systole, it will squeeze, means contraction and relaxation. Diastole means relaxation, and systole means contraction, lub dub like that. Okay, so this is the whole process. Normally this whole process needs some, like what they'll say, some things or activators or regulators to go on. So here you can see that simply to differentiate the diastole and systole, in the diastole the walls are open, whereas in the systole the valves are closed, means the blood flow some for 1 millisecond it will stop, means the valves will close and open, and that the blood flows through, like close in the opening process the blood is like the deoxygenated blood which enters the heart is oxygenated in this process. Okay, so like that the blood will flow throughout the body. Okay, so here they have given some of the regulatory things that will regulate the cardiac activities of the heart. First one is nodal tissues, like you can also say the pacemakers, and second one is special neural center in the medulla oblongata that is present within the heart, and adrenal medullary hormones and adrenal cortical hormones. So in these four options you have to say which one are responsible for the regulation actually. So if we see the chart here, you can see that normally the four options means both the four are responsible for the heart activities, but each one have the different role. For example, nodal tissues or pacemakers, what they'll do, they'll initiate the heartbeats. Okay, and medulla oblongata, it will regulate the cardiac activities via the nerves, means if you have the heart here, it will regulate the cardiac activities with the nerve connections. Okay, and adrenal medulla hormones which were released, what they'll do, they'll modify the heart rate, means normally we know that 72 bpm per second, right? So it will modify the heart rate according to the blood pressure, like what they'll say blood flow actually. Okay, blood flow, according to the blood flow it will modify the heart rate, whereas the adrenal cortex hormones, they will affect the blood pressure, not direct control, means both of them are responsible for the regulation, but in that these three are mainly responsible for the cardiac cycle. Okay, you can say that this nodular tissue, adrenal medulla hormones and medulla oblongata are responsible for cardiac cycle. Okay, but this one is not responsible for like it will not regulate the cardiac cycle. What it will do means it will affect the blood pressure, so it does not play a major role in the cardiac cycle. So that's why what are the main options from that question are now A, B and C, means nodal tissues, medulla oblongata and adrenal medulla hormones are responsible for the cardiac cycle here. Okay, we'll move on to the next one now. So what is the next one? Yeah, which of the following organisms cannot fix nitrogen? So it came from, like you have to say which bacterium or organisms, microorganisms will not fix the nitrogen they are asking. We know that there are some nitrogen fixing bacteria are present in the environment. So in these four you have to say which one is not responsible. So by this diagram you can easily understand that the wall works normally. So these are all the bacteria which can fix the like nitrogen actually. Azotobacter, Anabina, Nostoc and Oscillatoria, these are all the bacteria actually, they can fix the nitrogen in the form of some of them in ammonia or nitrous oxide, like that. Okay, normally in the form of ammonia. Okay, so whereas the volvox, it is not a, like it is like a flagella, it's not a bacterium. Like what it will do means it is like an algae actually. Volvox is an algae, it is a green colored algae. Here you can see the structure of the volvox here, like it will present. So if you see that there are two types of volvox actually. This is the volvox colony and this is the single volvox here. So what it will do, normally the green algae will not, this volvox will not fix the nitrogen. What it will do means, like this is responsible for some of the photosynthesis process in the algae itself, like, but normally means it can perform the photosynthesis but it lacks some enzymes like nitrogenase. That's why it cannot fix the nitrogen that was observed from the water itself. Okay, if you see the volvox structure here, you can see the flagellae with which it can flow or move. Okay, this is the whole colony of the volvox and these are all the flagellae of that volvox. Okay, normally you can say that algae means nachu in Telugu. Nachu అనేది ఎలా ఉంటుంది, like it is like a layer, right? So like that only you can see the algae in this form. Okay, it appears in the colonies itself and it also contains its own nucleus, chloroplast. That's why it can perform the photosynthesis. Okay, and this is the important vacuoles. Obviously every organism contain vacuole. Why? Because it have to store the food for next, like for the further life cycles. That's why they contain the vacuoles only. Okay, so here the Volvox is the answer, which cannot fix the nitrogen. Why? Because it lack nitrogenase enzyme, means it lacks the nitrogenase enzyme. Okay, and structures like heterocysts, which is needed for the nitrogen fixation. That's why it cannot fix the nitrogen. Okay, we'll move on to the next question. What is the next question? Is given below are two statements. In this you have to say which one is correct, which one is wrong, and both of them are correct or not. So these questions are given from the genetics chapter actually. So transfer RNAs, it belongs to the RNAs actually, the type of RNAs and everything. If you see the structure here, so this is the RNA structures. Like if you see it, what is that means? The transfer RNA and ribosomal RNA do not interact with the mitochondrial RNA. First statement. Okay, the RNA interference takes place in all eukaryotic organisms as a method of cellular defense. Means here they are asking that there is any connection between the tRNA or rRNA and mRNA. Okay, in the first statement, so we know that normally here if you see that, so these are the tRNA, and next one, this is the mRNA. Okay, so ribosomal RNA is normally present within the ribosomes. Okay, if you see, like I'm drawing here, so this is the ribosomal RNA, it is present within the ribosomes, so you cannot see in the life cycle. Okay, if you cannot see in the cycle here. So if you see here, this is like a translation process. You can see that the messenger RNA is passing through the ribosomes, and then the tRNA is carrying the codons, and it is coding the coding the messenger RNA, and it is forming the anticodons to produce a peptide or protein. Okay, so here first according to the first statement, you can say that transfer RNAs and ribosomal RNAs do not interact with mRNA. Normally we know that the transfer RNAs interact with the mRNA by the structure. You can say why? Because with their interaction only we can produce the proteins through the translation process. Okay, so in this you can say with the transfer RNAs only, this is the wrong, and along with the ribosomal RNA also interacts with the mRNA, how we will see. And in the second option they are asking, so the statement 1 is wrong here. If we see that statement 2, RNA interference means RNA takes place in all eukaryotic organisms. Okay, so you can say that RNA will interact with the eukaryotic organisms as a method of cellular defense they are asking. So if you see here, normally that the rRNA is also responsible for everything, and RNA interference takes place in eukaryotic organisms. Why? Because they are helpful so much for the cellular defense. So option 1 is correct here. Okay, we'll move on to the next question. Sorry, not option 1. The statement 1 is wrong, and statement 2 is correct here. Okay, so in the previous question, if you move on to the next question, what is the next question here is, in the represented plasmid, an alien piece of DNA is inserted at ECO-R1 site. Which of the following statements will be chosen to select the recombinant colonies? Actually, they have given the picture like this in the question paper. I have analyzed this and I have given in steps actually. So if you see it in this picture, we can see that there are so many. This is a alien piece of DNA. Like you can see that this is a bacterial organism which contains all the enzymes and everything. Here you can see that beta galactosidase is present and ECO-R1 site is present. BAM, H1, ORI, ampicillin, everything are added to that sample. Okay, if you see this sample, if that alien in this area, what they are saying that they'll add the DNA in the place of ECO-R1 site. So what will happen if they add some amount of DNA into this ECO-R1 site which is containing the beta galactosidase enzyme? Okay, if you see here, what will happen normally, here they have given the options like using ampicillin and tetracycline containing medium plate, and you have to choose which one they are asking. Using ampicillin and tetracycline containing medium plate, and blue color colonies will be selected, white color colonies will be selected, blue color colonies grow on ampicillin plates can be selected. Okay, here you have to say that which one option is right. Here in this you can say ampicillin and tetracycline and beta galactosidase all there, but according to this question, the ampicillin and tetracycline are not related to this answer. Why? Because the gene is like the DNA is attached to the ECO-R1 site of here, right? if you see that it is attached in the eco-R1 site where the beta galactosidase enzyme is present. So what will happen means, normally the beta galactosidase enzyme, what it will do means, in the presence of the beta galactosidase enzymes, when the DNA is inserted, they are not active. Okay, you can say that they are like in the presence of this, they are not active. The DNA, if it is not inserted, then only the beta galactosidase is active, and so the enzyme functions very well. So that you can see the blue color colonies normally. In the tetra plate here, when the DNA is not inserted, here you can see the blue color colonies in the tetra plate. Whereas when we add the DNA sample to this tetra plate, obviously what will happen means, this DNA, this recombinant DNA, what it will do, it will inactivate the beta galactosidase activity, and then the colonies, the blue color pigment which is released from this beta galactosidase chromogenic segment, actually this beta galactosidase or protein, it contains a chromogenic substance which will emit the blue color. So what will happen means, that chromogenic substance is deactivated, so that the colonies will turn actually white color, means colorless, actually means this DNA will deactivate that beta galactosidase enzyme when it is attached here. So obviously the colonies were deactivated. Okay, so like that it will happen. So according to this you can select the answer. So when it is added, obviously what is the correct answer here is white colonies can be selected. Why? Because when we add the DNA, we can see the white colonies. So that white colony is only shown the presence of this recombinant DNA. So this is the right answer. If we move on to the next one, so what is the next question is, which of the following genetically engineered organisms were used by Eli Lilly to prepare the human insulin? Okay, here they are asking about the recombinant DNA technology, what that was performed by Eli Lilly. Okay, company. If what they have done means, normally they have produced the human insulin and they named it as a humulin. Actually the human insulin is known as humulin. It was produced genetically by using the recombinant DNA. Okay, so in this process you can see that, so they have taken some like a phase. Okay, normally what the scientists did in that Eli Lilly company means, they have taken the human insulin gene into the plasmid. So here you can see the human insulin subunit here, this purple one. Okay, you can see the two differences, human insulin A subunit and B subunit, two samples they have taken. In that, this human insulin subunit is introduced into a plasmid of a bacteria. Okay, so normally the bacteria they have taken is nothing but the E. coli bacteria. So they have introduced this insulin into this plasmid, and then they inserted this plasmid into the E. coli bacteria. Okay, so they have seen that two types of insulin they have taken. No, each one is containing, this plasmid contains the promoter gene and antibiotic resistance gene and beta galactosidase gene, obviously. So you can see that these recombinant colonies, both of them have shown the different colors. Why? Because they have taken the different units, B subunit and A subunit. Okay, so what happened means, in the result they purified the fusion proteins that have produced from these recombinant DNAs. Here you can see that they have seen the colonies. Why? Because when this bacterial DNA combines with this human insulin, they have produced the color of the insulin protein. So by this recombination process, they have selected the colonies according to the color that appeared on the tetraplate. So colony 1 appeared the purple color, as the colony 2 appeared the green color. Why? Because it contains the A gene and it contains the B gene here. So like that they have produced the human insulin through the recombinant DNA in a higher amount. Okay, normally what is the use of recombinant DNA technology? We can produce the more amount of DNAs which were required for us, right? So in the higher amount they have produced it by using the E. coli bacterium in the lab. Okay, so they named that human insulin into, actually this human insulin into humulin. Okay, so this is the protein they have produced from the bacteria. So what they are asking, which organism is used for this whole process? They have taken the bacteria. So obviously the option 1 is right here. So they have taken the bacteria for the insertion of the plasmids here and production of human insulin. Okay, we'll move on to the next question. So what is the next question is, name the class of enzyme that usually catalyzes the following reaction. Okay, here they have given some enzymatic reactions or normal chemical reaction. In this you can see that SGSU. So SG means nothing but this is a normal chemical reaction. S means substrate and Z means a functional group other than hydrogen. In this reaction they have saying that this functional group is other than hydrogen. So when this substrate containing the functional group combines, like reacts with the second substrate, S2 means second substrate, obviously what happened in the chemical reaction means, so this functional group is transferred to the second substrate and it formed a complex with the second substrate, leaving the first substrate. Okay, so this is the chemical reaction. In this, so what they are asking means you have to say that the enzyme which participated in this chemical reaction. Okay, which induced this chemical reaction, they are asking about the enzyme here. They have given the 4 options: hydrolases, lyases, transferases and ligases. So if you see here, normally in the picture, simply I am saying it. So if you see here, so this is substrate 1 or substrate, you can say that substrate. What happened is, this is the substrate 1 actually, okay, S1, and this one this peptide is S2, which is like sorry this one is F1. 1 minute. So this one is S1 here. So this substrate, this fluorescent peptide is F1 here. So this is the G group here, like the fluorescence you can see. Okay, so this lipid is the S2 here. Okay, S1 or S2. So what happened when this any lipid combines with the fluorescent peptide, it undergoes a reaction in the presence of transferase enzymes. Transferase, why they named as transferase means here the functional group is transferred from one substrate to the another, right? So in the when the chemical reaction was taken, what happened in this reaction means, here you can see that this fluorescent protein containing the peptide is attached to this lipid. Okay, so like that you can see the formation of substrate 2 with the functional group. So all this happened with the help of the enzyme, that is nothing but the transferase here. So transferase is the enzyme which transfers the functional groups from one substrate to another. Okay, this is the whole process which takes place. We'll move on to the next question. What is the next question is, find the statement that is not correct with regard to, okay, here they are asking about the monocot stems. We know that there are two types of stems, monocot and dicots, right? So here if you see the picture of the monocots, here you can see that there is a epidermis layer, hypodermis, ground tissue and vascular bundles. This is the simple structure of a monocot. So here they are asking, they are asking about which is not correct about the monocot stem. Okay, so they have given the options here. What were the options were, if you see, hypodermis is parenchymatous. Here they are saying that the hypodermis is parenchymatous, or vascular bundles are scattered, and but the third option is vascular bundles are conjoint and closed, and the phloem parenchyma is absent. Okay, so in these 4 you have to say that which one is not correct. Okay, which one is wrong. So if you see here, if you see the structure here, vascular, first of all, hypodermis is parenchymatous, they are saying, it is not parenchymatous actually. You can see that parenchymatous structures have different things. So here the wall of the monocot stem appears like this, like in the like thin, like in the form of fibers which were having pointed walls and secondary walls and lumen. They'll appear like this in the form of narrow walls and everything. So normally parenchymatous tissue will not appear like this. So we'll see which type of tissue is it. If you move on to the second option, vascular bundles are scattered. Obviously here you can see that the vascular bundles are scattered around the inside the cell. okay, inside the stem. So this is the right option. And vascular bundles are conjoined and closed, right? Only they are closed only and conjoined with each other. And phloem parenchyma is absent. Normally phloem parenchyma is absent. This is right. Why? Because here you cannot see the phloem parenchyma in the monocot stem. So here in these options, these three are right. So hypodermis is parenchymatous is wrong. So we'll know that hypodermis is which type of tissue it contains. It contains the sclerenchymatous tissue actually. So sclerenchymatous tissue normally appears like in arrow bundles, and it shows the thick secondary wall and some type of lumen inside it, and it appears like this. So this the hypodermis of the monocot stem is obviously sclerenchymatous. So this is the right answer. The first one, sorry, like here the option 1 is wrong, means it is sclerenchymatous, means the hypodermis is sclerenchymatous here. Okay, we'll move on to the next one. What is the next question is the correct sequence of events in the life cycle of bryophytes. Okay, so another thing, this one also actually, yeah, in this session, I forgot to tell you that questions that were covered in our previous PYQ session, this one is also covered, and another two questions are all there. We'll see it. So, if you see the life cycle of your bryophytes, like the pteridophytes, they'll have, they are also having all these things. So, if you see here, normally what they'll do, they'll produce the antheridia or egg. So here they have given the options here. First one is fusion of antheridium with egg, attachment of gametophyte to the substratum, and reduction division to produce the haploid spores, formation of sporophyte, release of anthroids into the water. So you have to arrange them in order. Okay, here they have given the 4 options. So we will see the options here. First of all, what is the thing that was happening in this bryophyte cell is, if you see the structure, okay, first one is nothing but the attachment of gametophyte to the substratum. Okay, you can see that the gametophyte is attached here. The gametophytes are attached to the substratum. So this is the first step that was taken in the life cycle, that was undergone in the life cycle of the bryophytes. So after that, what will happen? Obviously, the release of, when these gametophytes are under and like attached to the substratum, they'll produce some antheridium in that antheridia are released into the water here. So this is the second step, and then antheridia or anthrozoids are released into the water here. And third step, what will happen? These released antheridia, what it will do, they'll combine with the egg. Okay, they'll combine with the female gametophyte, gamete and egg in the presence of water. Okay, this is the third option. And fourth option is obvious, when they combine with the egg, what they'll do, they'll form a mature sporophyte by the fertilization, like through the process. Okay, through this process, like fertilization process, they'll produce the sporophyte, and then that sporophytes again show the reduction division to produce the spores which were released into the environment. Okay, this is the whole process that takes place in the bryophyte cycle. So here, according to the options, you can say that the first one is obviously be the attachment of gametophyte to the substratum, and then option E, release of the anthrozoids into the water, and then that released anthrozoids will obviously fuse with the egg, and then when they combined with the egg, you can see the formation of sporophytes. These sporophytes, they again go on with the reduction division to produce the spores. So what is the order here? B, E, A, D, C. So in this you can say that the third one is the right answer here. Okay, so, and next one is nothing but it came from the CT scan and MRI scan that belongs to the tumor cells. Okay, this question they have given the 4 options. You have to say which one is correct here. Okay, what is the first option here is the computed tomography, nothing but CT and magnetic resonance imaging defect, means like MRI detect cancer of internal organs. So they are saying that they'll detect the cancer of internal organs. First option. Second option, the chemotherapeutic drugs are used to kill the non-cancerous cells. Okay. Third option, alpha interferons activate the cancer patient's immune system and help in destroying the tumor. Fourth option is chemotherapeutic drugs or biological response modifiers. Fifth option is in the case of leukemia blood cells, counts are decreased. Okay, in the case of leukemia, blood cell counts are decreased. Okay, in these options you have to say which one are correct. If you see that first option, normally the computer tomography and magnetic resonance image, obviously they'll detect the internal organs. If you see that this is the CT scan and MRI scan of your tumor patient, in this you can see these red spots are the tumors that were there in the CT scan of your brain, and this is the tumor that was formed in the CT MRI scan. Okay, so you can say that the CT scans and they can obviously detect the cancer cells and everything like internal organs which were affected by the cancer. And second option is like chemotherapeutic drugs are used to kill non-cancerous cells. Here this is the wrong one. Why? Because chemotherapy is normally done for the cancerous patients, so they'll not use it to kill the non-cancerous cells, they are used to kill the cancer cells. Okay, so this is the wrong option. Third option is alpha interferons activate the cancer patient's immune system and helps in destroying the tumor. So normally this is the right answer. The alpha interferons are the substances which will activate the cancer patient's immune system, means they'll increase the immune system of the cancer patients and they'll help in destroying the tumor. And fourth option is chemotherapeutic drugs or biological response, they are not biological response modifiers actually. Okay, in the case of leukemia, blood cell counts are decreased, they are not like decreased actually. This is not the right answer. So according to this you can say that what is the right answer here is A and C only. Why? Because CT scan and alpha interferons, like these two are good options, like right options from this question. Okay. So here you can also see that I have given one example how the alpha interferons will affect the tumor cells. Okay, so the tumor cells, like if you see here, these are the immune cells, the NK cells and T cells are nothing but the immune cells, we know that. So when they, so this is the interferon proteins. Okay, interferon A and interferon alpha and interferon beta proteins, how they'll do means they'll enter every part, like they can kill any tumor cells present in the brain or liver or everything. What they'll do means these interferons, when they enter the tumor cells, they'll decrease the cell count of the tumor cells actually. So the alpha interferons, they'll activate the immune system and help in destroying the tumor cells by decreasing the cell count, means they'll not allow the tumor cells to divide. Okay, they'll stop the division of the tumor cells, like that they'll decrease the cancerous agents to cause cancer. Okay, that's it guys. So we'll move on to the next video in the next session. In this way, in that we'll cover the remaining questions that came from the biology paper. Okay, like for example, these are the questions that came from the biology paper in the next session. Thank you guys. I hope all of you like the video, and if you like it, just click the like button, and if you thought that this video is helpful for you, just share it with your friends. And if you want to watch the more videos or more information from the Biosastra Talks, just subscribe to my channel Biosastra Talks. Okay, so I hope you'll wait for the next video, and we'll